are going towards 2D electrophoresis, and this two-dimensional electrophoresis or second-dimensional electrophoresis. So choosing a length of IEF, Isoelectric Focusing Strip. So how much of the length should be preferred. So as the length of the strip, actually we are talking about strip because we will be using this in two-dimensional electrophoresis. So the length of the strip increases when it increases the loading capacity. It increases the loading capacity. It increases the resolution of proteins. So with increasing the length, the resolution of protein increases. The number of spots detected means this is the output. This if the figure in the right top panel, that is this is the output, and these spots increases if the length of the strip actually increases. And next is focusing time increases. And cost effectiveness decreases, means it will cost us much if we increase the length of the strip, because it will be costly then. So the different lengths of the strip are there: 7 centimeter, 11 centimeter. Normally people do prefer 11 centimeter, but 7, 11, 17, 18, 24. These are the different types of the different lengths available for the IEF strip. And these strips are of different, they have different capacity, like this is plus 3 to 10, plus 5 to 8, plus 4.7 to 5.9, and so on. So these are different. So these are ready strip. IPG strips are pre-printed to indicate anode. एक साइड पे इसका anode लिखा होगा, second side पे इसका negative, एक side पे plus होगा. And the pH range, also the pH range is shown. And in addition, a barcode is printed on the 24 centimeter strip, So here it is shown. A barcode is also there. So this is the general information about the strip. And this, if you can see the strip, this strip is the indication. This is pH 3, and this end will be that will be pH 10. And here, this is the anode, plus sign, and this is the cathode. And actually this is wrapped in a parafilm. So while using this, the parafilm is removed, and now this can be used for electrophoresis. So this is all about the electrophoresis, the isoelectric focusing strip actually. जी, क्लास समझ आ रही है?
Yes sir, आ रही है.
इजी है?
Yes sir.
अच्छा, प्रैक्टिकल में ये किया हुआ कि नहीं? You have also practicals. So are you done with two-dimensional?
No sir.
Sir online में किया हुआ ना, वैसे तो नहीं किया.
ऑनलाइन में two-dimensional किया हुआ है अनाया?
No
No
sir.
sir, नहीं किया हुआ.
No sir, अभी तक नहीं किया.
Okay, so now like first step is to select the strip for two-dimensional gel electrophoresis. ठीक है? So strip selection है. उसके बाद हमने देखना है कि हम इससे पहले पढ़ चुके हैं कि which pH range should be preferred for our proteins. ठीक है? So we have covered that before, and now the length of the strip, and next to it is, so who will explain this slide? Like who will explain so that we can proceed further?
Sir मैं कर दूं.
जी अनाया, please.
सर आइसोइलेक्ट्रिक फोकसिंग स्ट्रिप की लेंथ मैटर करती होती है, क्योंकि अगर लेंथ इंक्रीज करते हैं तो उसकी लोडिंग कैपेसिटी, जो प्रोटीन को लोड करते हैं, लोडिंग कैपेसिटी इंक्रीज होती है, रेजोल्यूशन ऑफ प्रोटीन इंक्रीज होती है, नंबर ऑफ स्पॉट्स डिटेक्टेड वो भी इंक्रीज होता है, और फोकसिंग टाइम भी इंक्रीज होता है. लेकिन ये कॉस्ट इफेक्टिव होती है. जितनी ज़्यादा लेंथ होगी उतनी ज़्यादा उसकी कॉस्ट होगी. और ये डिफरेंट रेंजेस में आती है, 7 सेंटीमीटर, 11 सेंटीमीटर, 17, 18 एंड 24। और उसके ऊपर डिफरेंट जो ये इसकी पीएच की भी डिफरेंट रेंजेस होती है, जिसमें ये होती है +3 to 10, +5 to 8। और इसका जो ये होती है, इसकी एक साइड पे हमारे पास एनोड होता है जिसकी पीएच 3 होती है, और जबकि दूसरी साइड पे कैथोड होता है, उसकी पीएच 10 होती है। ये पैराफिल्म के अंदर रैप होती है, और उसके बाद हम उसको उसमें डालते हैं।
ठीक है, that's good. Okay, and if you can see, before this we have electrophoresis. Those were termed as one-dimensional, and those were like what will be the difference in two-dimensional? This will be like this. So in the first dimension, it will move in one direction, and in the second, so now the proteins are resolved. We will be having different spots like this. We will be having different spots. One spots will be there, and these spots will be like, and if this spot, if say for instance, if this spot has multiple proteins, and they converge at this site, so there is another dimension, and this whole gel will be moved in this direction. So at this very spot we will be having like 3 or more spots here at same position. So maybe 2 spots here, maybe 2 spots here. So in this way, maybe 1 spot here. So in this way, the 2 dimensional. So now this will be the result now. This is the result now. Before this, in 1 dimensional, this was the result. This was the result, but now this is the result. Am I clear?
Yes, sir.
And so this is important and most used in day-to-day experiments in proteomics actually, and basically, and to understand this we have a video, and after checking that video we will proceed further inshallah.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the Ipg strip. The second segment will be on how to equilibrate the Ipg strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the Ipg strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 Ipg strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has two sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before this step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode with the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I opened the gel cassette, I used forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kumasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kumasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kumasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kumasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
And that's done. So next to it is so class समझ आ गई है?
Yes sir. आगे है.
Yes, sir.
Am I clear?
Yes sir.
ठीक है, सही है. अच्छा, कलीम समझ आ गई? कलीमुल्लाह
यस सर,
समझ आ गई कि
मेरे
नहीं?
थोड़ी बहुत है क्योंकि सर मेरे पास वो नेट प्रॉब्लम था ना, वीडियो रुक जाती थी इसलिए.
चले चले इस वीडियो को आपने देखना है फिर, ठीक है, और
यस सर.
बताना है. अच्छा, फरा आपको समझ आई है? फरा नदीम.
यस सर, वो नेट स्लो था, अभी से इतना नहीं सुना था मैंने.
अच्छा, एंड शुएब समझ आई कि नहीं? अजीम इज देयर? अजीम
यस सर, एक दफा दोबारा फिर देखूंगा, मजीद कुछ चीजें रह गई थी.
हाँ, इसमें पूरी क्लास के अजीम आपने अच्छा पॉइंट रेज किया. इसमें बहुत सारे क्वेश्चंस रेज होंगे, आपने इसको गौर से देखना है, वहां पे ही आपको आंसर मिल जाएंगे. लाइक इन्होंने एक बात याद रखें कि इन्होंने जो किया है पहले, जो है ये जो जेल बनाने का तरीका है, नीचे एक्राइल एमाइड जेल है, हम प्रोटीन्स के लिए एक्राइल एमाइड जेल यूज कर रहे हैं. फिर वो जो स्ट्रिप इन्होंने रन किया था पहले, इलेक्ट्रोड वो करके तो रन किया था उसको, आइसोइलेक्ट्रिक फोकसिंग वाली स्ट्रिप जो है, उसको जब इन्होंने रन किया, उन्होंने वो क्या किया, इन्होंने एगरोज ये एक्राइलेमाइड जेल के ऊपर रखा स्ट्रिप को, और उसके ऊपर ओवरले द एगरोज ओवरले वाज यूज्ड. सो नाउ दीज आर टू टाइप्स ऑफ जेल्स. नाउ द फर्स्ट वन वाज एक्राइलेमाइड जेल एंड द ओवरले वाज द एगरोज जेल, एंड वन इज द मार्कर. एक वेल इन्होंने मार्कर के लिए भी रखा है कि जिसमें मार्कर को ये फिर रन कर सके, जो छोटी सी एक स्ट्रिप ली है मार्कर की. सो आपने like you have to go through this video again and again. You will have question in mind and you can address that if still you have the question, so we can discuss in the coming class as well. Am I clear?
Yes sir. Sir,
Yes,
एग
sir.
रोज एज अ लोडिंग जेल यूज किए थे.
नहीं, हाँ, एगरोज को इन्होंने स्ट्रिप को तो एक रोज उस पर रखा है ना, जेल के ऊपर, उसके ऊपर एगरोज का जो है ना एक ओवरले बना दिया था कि वो फिक्स रहे अपनी जगह पे, और फिर जब रन करेंगे इसको तो वो फिक्स रहे सारी चीजें. ठीक है, सो वो एज ए लोडिंग उसके लिए यूज की थी, जगह के लिए, ताकि वो
सर
सपोर्ट प्रोवाइड करें. जी जी.
सर, रन करने के बाद भी जो भी हमारा प्रोटीन का सैंपल होगा सर, वो एग्जैक्ट उसी प्लेस पे ही आएगा जिस प्लेस पे उसे आना चाहिए था, अकॉर्डिंग टू इट्स साइज.
क्या मतलब? आपका क्वेश्चन मैं नहीं समझा.
सर, हम लोगों ने इसमें दो तरीके की यूज किए ना, हम लोगों ने 2D किया ना इलेक्ट्रोफोरेसिस.
हाँ,
सर
बिल्कुल
मैं
बिल्कुल.
कह रही हूँ
जी
मैं कह
जी.
रही हूँ अगर जब सैंपल हमारा जेल के ऊपर रन होगा, स्ट्रिप से उसने ट्रांसफर होना है ना जेल पे, तो कहीं ऐसा तो नहीं होगा कि बीच में सैंपल की थोड़ा सा मतलब ओरिएंटेशन थोड़ी सी चेंज हो जाए.
नहीं, वो तो बिल्कुल उसी डायरेक्शन में जाएगा, प्रोटीन ने उसी डायरेक्शन में जाना है, मींस दैट वेल टुवर्ड्स द ग्रेविटी. ठीक है, और पहले वाला जो था ना, वो तो हॉरिजॉन्टली जा रहा था, एंड दैट वाज ऑन आइसोइलेक्ट्रिक फोकसिंग. ठीक है, तो
यस
वो
सर.
बिल्कुल भी अपने लाइक जहां पे जिस तरह पहले हमने ड्रॉ किया था ना, लाइक जिस डायरेक्शन में प्रोटीन ने फर्स्ट इस तरह डायरेक्शन में जा रही थी ना प्रोटीन, और फॉर
यस
इंस्टेंस
सर.
यहाँ पे फॉर इंस्टेंस बैंड्स हैं, द बैंड्स आर लाइक हियर, यहाँ पे एक बैंड है. सो इस बैंड को, इसको फिर जब इस डायरेक्शन में रन करेंगे, सेकंड डायरेक्शन में, सो अगर जब इस डायरेक्शन में इसको दिस वन इज अ स्ट्रिप, so this one was the strip. सो इस स्ट्रिप को
Yes.
जब इस डायरेक्शन में रन करेंगे सो वो क्या होगा कि लाइक द बैंड, अब इसके अंदर बहुत सारे बैंड मौजूद होंगे जो इस जगह पे आए हुए हैं. सो दिस बैंड विल बी रिजॉल्व लाइक दिस. यहाँ पे एक बैंड आ जाना है, यहाँ पे एक आ जाना है, बिकॉज नाउ दिस इज नाउ इन द अदर डायमेंशन, लेकिन ये एक ही उसमें होंगे. अगर यहाँ पे कोई बैंड है सो और अगर एक ही है सो दैट विल बी देयर. और अगर इसमें मल्टीपल बैंड्स हैं सो लाइक एक यहाँ पे है, दूसरा यहाँ पे है, सो ये एक ही साइज पे होंगे. तीसरा बैंड जो है वो इफ दैट इज लाइक हियर सो इस तरह आएंगे वो. ठीक है, एम आई क्लियर?
Yes, sir.
तो जी
Yes,
जी
sir.
जी जी ठीक हो गया. सो आफ्टर दैट we have so basically in isoelectric focusing, so the vertical slab gel like SDS-PAGE are capillary, and the horizontal can be liquid ampholites or strips, that is for example in 2D page that are used. So isoelectric focusing, so isme vertical can be used, horizontal can be used for isoelectric focusing. And if you can see this one, this is from pH 9 to pH 3, and that is linearly decreasing pH. So the pH is decreasing. So if you can see this one, this diagram, so that is from the negative to the positive, from the cathode to anode, and the protein will move. So sample loading will be here. So there is no sample. This is only the gel. The sample loading is there, and now this is resolving. The gel after protein staining. So after running and after staining, it will, the different bands will be there, they are visible. And if you can see this one, the upper panel, this is from like from 10 or 9 to 3 pH. And if this strip will have like that, they are all at high pH, the protein is negatively charged. This negatively charged protein will move toward anode. This will move toward the positive, and the positive charge will move towards this direction. And if they have like they have a certain charge, and so they will move like they will come at a certain point, at isoelectric point. So that they will adjust like here. Some of the protein will adjust like some other where. So some like something like in another place. So after that, the steps in horizontal IEF, like isoelectric focusing. So place, so if we are doing with horizontal IEF, so place a few drops of like light parafilm oil to adhere the plastic template to the cooling plate. So the cooling plate, and smear it over the surface. So and next to it is place the template on the cooling plate, and make sure there is no bubble trapped. We have already like checked in the videos as well. So avoid any bubble formation in the between the cooling pad and the template. And next is place a few drop. Okay, so align the gel carefully on the template, again ensuring that there is no bubble. So this is done. So press the wrapping of the gel to squeeze out air bubbles. And when there is no bubble, remove the wrapping entirely. And after that, the applicators, sample applicators follow like filter paper, using a pair of forceps on the gel. So these are once the applicator, so here the samples can be applied. So once the applicator has been placed on the gel, do not move it. So load 15 microliter sample. So this can vary to 15 microliter of the sample on the applicator with a micro pipette. Load the same amount of standard on the first, middle, and the last applicators, and soak the electrode strip in the appropriate buffer, like cathodic strip 1 molar sodium hydroxide, anodic strip so 1 molar this H3PO4 and phosphoric acid, and lay the strip at the designated position on the gel. So the technique changes with every different setup. Like the equipment is different, so the techniques differ a bit, but the concept is same. unscrew the electrode holders and slide them up till, so this is another equipment actually, so, and slide them until they are over the electrode strip, then place the electrode plate over the gel, place the safety lid back on the on the equipment, and so these are the dry strips. These are the dry strips as has been shown in the video. You already have checked this. So these strips are used for 2D electrophoresis and dehydrated gel between two plastic wraps. So gel needs to be rehydrated. So the dehydrated gels needs to be rehydrated. So contains immobilized ampholytes for defined pH ranges. So and different lengths are available. Samples is applied on the gel face down and are by cup loading. So this is all about this. So cup loading and the other, like the high voltage, and this is the low voltage. So we have covered some of these things in the video, and we will be also discussing all these things in the coming lecture as well. And if you can see choice of pH range and length, so you can see the guidelines for choosing immobiline dry strip gels, and you can see the range of the pH range. So this will be the pH range, and the strips available are like this. So if you want to have a narrow pH for your protein of choice, so the strips available you have are like this. At this range, only two lengths of the strips are there, so the equipment will change if the you have different, like if you have this length and now you want to run this length, so you will have to change the equipment or the consumables. And here different pH, these are the different pH range, and these are different lengths.
So basically, we were talking about like two types of the isoelectric focusing. The first one was vertical slab gel, two types of gels actually. The slab one, and we have already talked about SDS-PAGE or capillary, and the second one is horizontal and can be like liquid ampholytes that contain liquid ampholytes or strips. And the strips one, this will be the 2D actually, the two-dimensional, and we already have talked about this and read about this in the video as well. Like the strip was used in horizontal and horizontally, and the rest of the procedure, the second dimensional, was actually the vertical one. And okay, so the first one was vertical and the second one was horizontal, and the horizontal may contain both of the vertical and horizontal actually. So next to it is like this is a linearly decreasing pH. If you can see this one, the capillary, and this one is high pH, the negative charge will be more, and if there is a low pH, the positive charge will be more, and the negative charge will move towards the anode, and the positive will move towards the cathode, and they will stop there when their isoelectric focusing point is reached. And if this is the gel loading, sample loading is there, and here the gel after protein staining, and that is the stained protein, the stained gel now here at this stage. And here is the representation of that is at high pH, the protein is negatively charged and will move towards the other pole, and from here at low pH, the protein is positively charged and it will move towards the other pole. And here there are the steps in horizontal isoelectric focusing, and that is actually the manual method. We have already checked that out in the video, the a bit automated one, and they place a few drops of like here what they do actually, they place few drops of paraffin
सर
oil.
आपकी आवाज बहुत हल्की आ रही
And wait for a while. So अभी ठीक आ रही है आवाज?
Yes, sir.
हाँ, so paraffin oil जो है वो like few drops of the paraffin oil is used to adhere the plastic template to the cooling pad. So this the below is the cooling pad actually, and that is and smear it over the surface. And rest of the things are clear, like to place the template on the cooling pad and make sure there is no bubble trapped between the cooling plate and the template actually. And rest of the steps are like align the gel carefully on the template, again ensuring that there are no bubbles trapped between the polyester backing of the gel. And here you can see from the pictures, so the care must be taken to avoid any bubble formation between the cooling pad, between the gel and the template actually. And okay, this is again the same thing. So remove the wrapping entirely after confirming there are no bubbles. And carefully place the sample applicators. So here the step of sample, the placing of the sample applicators, like they will be like filter papers. On these filter papers, sample must be poured on these filter papers, and after that they will be like they will move through isoelectric focusing. And there are the like lower 15 microliter of the sample on these applicators. You can see the white one. So the sample loading is gonna proceeding here, and load the same amount of standard on the first and the middle and the last applicators. So applicators. So we need also standards to confirm the movement of the sample actually, and soak the electrode strips in the appropriate buffer, like cathodic strip must be in 1 molar sodium hydroxide, and anodic strip must be in 1 molar like phosphoric acid. And rest of the things you already have done, and these are lay the strips at the design position on the gel. Excess length should be cut off so that the strips fit within the gel. So here you can see the strip, within the diagram, the strip has been like taken, and if the strip is lengthy, so you can cut that down to fit into the apparatus. And after that, unscrew the electrodes, slide them until they are over the electrode strips, then place the electrode over the gel and place the safety lid back, and now it's done. So these are the screws. The screws can be seen, electrode holders. The platinum wires inside should be over the electrode strips. And here, these are the dry strips. We have already observed this thing in the video as well. So they are of different sizes. So used for 2D electrophoresis, dehydrated gel between 2 plastic strips. So dehydrated gel between 2 plastic strips. So gel needs to be rehydrated. So it contains immobilized ampholytes for defined pH ranges, and different lengths are available. Different and sample is applied with gel faced down or by cup loading. So cup loading, we have the details in the coming slide for how the cup loading will work. So either we will apply the sample with the gel face down, this one, and we will apply the sample over there, or by cup loading we can do that. And here are the choice of pH range, like if you want specific pH or specific range of pH, which strip well we need actually. So these are the different strips, the sizes of different strips, the pH range, and there are different range of the pH, and there are different lengths of the pH of these strips actually, these dry strips. So in this range, All lengths are available, like 24 centimeter, 18 centimeter, 13 centimeter, 11 centimeter, and 7 centimeter. All ranges are available here. They are available here in this range from 3.5 to 4.5 pH range. Only 2 sizes are available. And rest of the things, they are common, like you can order any of the strip based on the confirmation, like what our desired protein is, like our desired protein range between 6 to 8.5, 6 to 9, or 6 to 9 centimeter, sorry, 6 to 9 pH. So we will order. Okay, so we will order this one instead of some other, because this do our work well, and we don't need lengthier, or we don't need extended pH range. So, and we can see which sizes are available, because if our apparatus is for 11 centimeter, and so our 18 centimeter will not work there, and this is available in 18 centimeter actually, and 24 centimeter, but our apparatus is for 11 centimeter, so this will not work for us. So we will choose, if our apparatus is for 11 centimeter, we will choose in this range, in this size actually. So this is how we need to confirm first the size of the strip and the pH range, our desirable pH range actually. And rest is okay for us. Like these dry strips, we have covered this already. How to load the sample on the strip, and then first uncoat the strip, and then load it in the tray, and then load the sample there. We already covered this in the video. And the passive rehydration gel strips are put face down over buffer plus sample 12 to 24 hours. It will take 12 to 24 hours for passive rehydration. And active rehydration, gel strips are put face down over buffer, sample, and low voltage. It's like it will need voltage, and is applied, and it will be from 12 to 15 hours. And cover fluid, cover fluid for and with parafilm or mineral oil, and it will prevent evaporation and drying. And next to it is the dry strips needs to be rehydrated with the rehydration buffer. And if the rehydration buffer is done without the sample, then it is usually passive, and the sample added by the cup loading. So cup loading can also, you can see the cup-like structure here, and this is movable actually. This movable cup-like structure, so the sample can be loaded here. And movable electrode is there, IPG strip holder, and you already know IPG, the immobilized pH gradient. So the electrolytes are actually immobilized. You know actually very well, even more than me, that what is meant by immobilized electrolytes. And washing with water, and place gel facing up in the strip holder, and then place a sample cup and test for leakage and rehydration buffer, remove rehydration buffer, add sample. And this is actually the procedure. We covered already these things. So anybody who could explain this? Kinza Majid, Kinza? शफीक, आपको आवाज आ रही है?
यस सर
जी, कैन यू एक्सप्लेन?
सर इसमें जो हमने आइसोलेटिव फोकसिंग की जो देखी, एक 2D होती है, एक वर्टिकल होती है। अब जो 2D यानी हॉरिजॉन्टल है, उसके अंदर हमने फर्स्ट देखा कि जो कूल पैड होता है, उसके ऊपर जब हमने अपने प्लास्टिक टेम्पलेट लगाना होता है, तो उसके लिए हम क्या करते हैं, हम पैराफिन ऑयल के कुछ ड्रॉप्स डालते हैं, ताकि वो ऊपर क्या नाम है सही तरह अधर हो जाए, और साथ इस चीज को श्योर करना होता है कि उसमें कोई बबल ना रह जाए। सेम थिंग उसके बाद उसी के ऊपर हमने क्या नाम है प्लास्टिक टेम्पलेट के ऊपर अपनी जेल प्लेस करनी होती है, एंड जेल के ऊपर फिर हमने छोटे छोटे जो सैंपल्स जो छोटी स्ट्रिप्स होती हैं वो रखनी होती है, जिसके ऊपर हमारा सैंपल होता है, जिसके अंदर हमारा सैंपल कंटेन होता है। तो ये माइक्रो सेंटीमीटर्स की स्ट्रिप्स होती हैं, अगर कोई लेंथी हो तो हम उसको काट के छोटा करके वहां पे प्लेस कर लेते हैं। और इसी तरह की इस सेम लेंथ की क्या नाम है जो होंगी यानी लेडर वाली सैंपल होंगी जो रेफरेंस सैंपल सेलेक्ट करेंगी उनके साथ, ताकि हम कंपेयर कर सकते हैं या उनको देख के हमें पता चल जाए कि हमारी जो ये हमारा क्या नाम है सैंपल रन हुआ है कि नहीं रन हुआ। इसके बाद जो सेकंड वाली थी उसमें जो टू डी स्ट्रिप्स थी, तो उनमें हमने देखा कि वो उनको डिहाइड्रेटेड होती हैं, वो उनको रिहाइड्रेट करने के लिए या तो कप लोडिंग करनी पड़ती है या तो फेस वाला मेथड यूज करना पड़ता है उनको रिहाइड्रेट करने के लिए। तो इसमें ये जो कप लोडिंग मेथड है इसमें हमने वो छोटे छोटे कप हैं उसके अंदर क्या नाम है हम अपना जो रिहाइड्रेशन बफर है वो लोड करते हैं। इसके अलावा जो स्ट्रिप है वो आईपीजी स्ट्रिप है, यानी इमोबलाइज पीएच ग्रेडियंट, उसके अंदर इमोबलाइज एम्फोलाइट्स की वजह से जो पीएच ग्रेडियंट है वो वहां पे स्टेब्लिश किया हुआ है।
ठीक हो गया, great. So, and next is we have a small video for this cup-like structure, like we didn't cover that before. So we have a small video for this as well.
To use sample loading cups with the Protean I-12, follow this setup procedure. First, place the rehydrated IPG strip gel side up in the focusing tray. At this point, you can add electrode wicks if you choose. Pre-wet the wicks and place them at the ends of the tray on top of the IPG strip. Next, insert the electrode assemblies into the tray. Match the positive electrode assembly to the positive side of the tray and the negative assembly to the negative side of the tray. They will only fit in one orientation because they have round and square pegs specific to each side of the tray and each electrode assembly. Press the electrode assemblies straight down into the tray by pressing on the green tabs until you hear a click. Place the tray on the platform of the instrument and insert the positive electrodes into the appropriate slots. Please note that the trays should be placed into the instrument according to the guides on the sides. The electrodes should be pressed into the instrument until a click is heard. Press the tray into the instrument until the stop on the tray is reached. Check to make sure that the pin on the negative electrode assembly has made contact with the metal strip on the instrument. Assemble the sample loading cup holder to hold as many cups as you would like to use for loading. The cups fit into the sample cup holder via arms on the sides of the cups that fit into holes on the holder. The holder is designed to hold the cups with a certain amount of give to allow a tight seal with the gel so that the sample doesn't leak and to ensure that the connection is not so tight as to damage the strip. Choose the desired location for the sample cup assembly. Snap the assembly onto the tray until a click is heard. Once the assembly is in place, do not slide it along the tray, as it will damage the IPG strip. If you would like to reposition it, unclip the assembly, pick it up, and reposition it on the strip. Add the sample to the sample cup. Overlay the strips and the sample within the cups with mineral oil. Close the lid, and you are now ready to program the cell. After the run is finished, the tray can be removed by sliding it away from the electrodes and lifting it out of the instrument. Remove the cup holder, and then the electrodes can be removed from the tray by pinching the green tabs and lifting up.
So that's up. So who will summarize the video? Fareen is there? Fareen?
सर
जी
सर सबसे पहले तो उसने रीहाइड्रेटेड स्ट्रिप जो थी सर वो अंदर रखी सर उसके बाद उसने वो इलेक्ट्रोड्स लगा दी
जी
और फिर सर उसके बाद वो असेंबली ट्रे थी जो उसने बताई कि आपने एक ही जगह पे रखनी है आपने उसको मूव नहीं करना वरना जो हमारी स्ट्रिप है वो डैमेज हो जाएगी तो सर उसके ऊपर उसने सैंपल लोड किया और फिर मिनरल वाटर किया था सर स्ट्रिप पर भी और सैंपल्स पर भी
मिनरल
और फिर
ऑयल
सर मिनरल ऑयल सर और फिर सर उसने भी ऑपरेटर्स में उसको रख दिया था रन करने
जी जी और इसके बाद उसने डिसअसेंबल किया और
यस
यही
सर सब
जो
कुछ
स्ट्रिप्स हैं जी जी इसके बाद फिर उसने 2D के लिए स्ट्रिप्स को रन करना है मींस के दिस वाज द फर्स्ट डायमेंशनल दिस वाज हॉरिजॉन्टल and this strip is actually the sample will move based on the isoelectric focusing and this was the first step of the 2D gel इसके बाद फिर दूसरा स्टेप है एंड दैट विल बी लाइक सिंपल हम इलेक्ट्रोफोरेसिस जो करते होते हैं लाइक बेस्ड ऑन द मॉलिक्यूलर वेट एक्चुअली ठीक है सो एंड उसके बाद स्टेनिंग So running conditions for IEF is for strip 50 macro angstrom and this is 10,000 volt generally typically start with low voltage we will start with low voltage and we will gradually increase through a series of steps to the desired focusing voltage so is more important so always start with low voltage. A long focusing time is required for long length of the strip, narrow pH range, if the pH range is narrow, so longer focusing time is required. Higher protein load, if the protein load is higher, so we need to run it more. Higher urea or detergent concentration, we need to run more. And okay, so who will explain this? Khansa is there, Khansa?
सर
जी बताएं दिस स्लाइड
सर आपने बताया है कि प्री स्ट्रिप्स की जो है वो इतनी होनी चाहिए मतलब 5 माइक्रो ऑप्शन से 10,000 की कर्नल इसमें हम आपने बताया कि हम सबसे पहले लो वोल्टेज से रन करते हैं फिर ग्रेजुअली उसको इंक्रीज करते हैं जब हमें लॉन्गर फोकसिंग टाइम रिक्वायर्ड होता है तो उसमें आपने ये हमें 4 पॉइंट्स बताए हैं जिसमें लॉन्गर लेंथ ऑफ स्ट्रिप्स नैरो मतलब पीएच रेंज जो है वो नैरो होती है प्रोटीन लोड जब ज्यादा हो और डिटर्जेंट कंसंट्रेशन हायर हो
जी इसका ये मतलब हुआ कि अगर स्ट्रिप लेंथ ज़्यादा है तो हमने फोकसिंग टाइम ज़्यादा रखना है ठीक है अगर पीएच रेंज हमारा कम है मींस के 3 to 4 है रादर देन 3 to 10 सो उसमें फिर हमने फोकसिंग टाइम ज़्यादा रखना है अगर प्रोटीन लोड ज़्यादा है हायर प्रोटीन लोड सो वी विल बी गिविंग मोर फोकसिंग टाइम and same is for detergent and urea concentration if they are more they are higher so we will need more focusing time. And next is this post IEF processing. So after IEF, so what we will do, if just doing IEF horizontal gels fix with the trichloroacetic acid and stain with the Comossi or silver staining. So silver staining just like SDS Comossi के साथ कर लें या silver staining कर लें. So that's it. For IPG immobilized pH gradient strips, staining with the crystal violet or zinc imidazole. If doing 2D pH, a strip can be stored at -80 for several months if not used immediately. When ready to second dimension must be equilibrated before an IPG dry strip can be used in SDS-PAGE. It has to be immersed in buffer containing SDS to allow focused proteins to interact with the SDS. So we need to interact the dry strip with the SDS solution first. And then long equilibration times, for instance, 10 to 15 minutes plus the use of urea and glycerol improve protein transfer to SDS page easily. So now, so we have what we have done. We have our protein on the strip. Now we need to transfer the protein because this will be on the strip and they will be and now we need to transfer the protein to onto the gel, and we need to run on the gel actually, and now we have separated based on the isoelectric focusing, and now we have to run these on the gel. So this will be the second dimensional now. So with that, before running that, we need to dip the dry strip into the onto the SDS into the SDS solution, and along with the if you use urea and glycerol, that will improve protein transfer to the SDS page. And next to it is this thing, like this one was the strip, and this is a separation in the first dimension, and this will be by charge, and this and the protein mixture are used, and the protein mixture are separated based on isoelectric focusing. And you can see here example is given that's from pH 4 to pH 10. And now apply the first gel on the top of the other gel. Now this is the SDS gel, this is the PAGE actually, polyacrylamide gel, and this is SDS. And now this strip is used to transfer the protein from the strip into the gel, onto the gel. And this is separation in the second dimension, and this will be by size. So this was initially by charge, and this is by size. And isoelectric focusing, this is also in this direction. This is actually horizontal, and now this is vertical. So next to it is you can see the results of the two-dimensional gel. And this one is, now you can see from basic to acidic, stable pH gradient, stable pH gradient here. If the protein are like here, they will be the stable pH gradient, and here they will be the proteins will be more basic towards here, they will be more acidic towards here. And you can see if we have run in this direction on the SDS, so this is the migration pattern. So the protein are migrated in this direction. So this will be a molecular weight into 10 raised to power minus 5, and this will be the SDS migration pattern. And so this is now the strip was like move the protein move like in this direction, from acidic to basic or from basic to acidic, or whatever the protein, the isoelectric point is. And after that, once that stabilize, we move those into the SDS gel, and like this is actually the result of the 2D gel actually. And these spots shows different proteins, and like here the proteins will be more basic. These proteins will be more basic. They will be at stable gradient. They will be in the middle, and they will be more acidic. And the difference between this and this protein bands, that means they have different molecular weights. ठीक है, कौन इसको explain करेगा? Who will explain? Come on.
सर मैं कर दूं?
जी शफीक।
सर इसमें हमने पहले आइसोइलेक्ट्रिक फोकसिंग के जरिए पीएच की बेस पे या चार्ज की बेस पे हमने क्या नाम अपनी प्रोटीन को सेपरेट कर लिया। अब उसके बाद हमने इसको मॉलिक्यूलर वेट पे सेपरेट करना है, तो उसके लिए हमें से जो 2D जेल है वहां पे अपनी जो पीएच स्ट्रिप है वहां से अपनी प्रोटीन सैंपल को 2D जेल के ऊपर ट्रांसफर करना है। तो उसके लिए हम एक तो एसडीएस यूज करेंगे ताकि जो प्रोटीन कन्फर्मेशन है वो स्ट्रेट लाइन हो जाए, जो नेट नेगेटिव चार्ज आ जाए इसके ऊपर, प्रोटीन के ऊपर। एंड सेकंड थिंग, हम उसके अंदर ग्लिसरॉल यूज करेंगे ताकि प्रोटीन अच्छी तरह रन कर जाए। तो इसी पिक्चर के अंदर जो उन्होंने दिखाया हुआ कि जो बेसिक पीएच और एसिडिक पीएच के दरमियान जो स्टेबल पीएच ग्रेडियंट है, उसके बेस के ऊपर चार्ज के ऊपर फर्स्ट तो प्रोटीन सेपरेट आउट होगी। अब उसके बाद हम इसको, अब जब उसके ऊपर एसडीएस यानी 2D जेल के ऊपर लेके जाएंगे, और वहां पे इलेक्ट्रिसिटी के दरमियान जब ये अपने मॉलिक्यूलर वेट के ऊपर फर्दर डिफरेंशिएट हो जाएगी।
ठीक हो गया, ठीक है। Okay, and next to it is like differential gel electrophoresis. And differential gel electrophoresis is DIGE. So differential gel electrophoresis has same principle as SDS-PAGE, and it can be done in 1 or 2 dimensions. Additionally, DIGE requires fluorescent protein stains up to 3 of the cyanines, a gel box, and a gel scanner. So we will need, this is just like SDS. This could be 1-dimensional or 2-dimensional. Additionally, this will require the stains. Normally, three fluorescent stains are used, and further, a gel box will be required, and a gel scanner will be required. So basically, 3 things are required: a gel scanner, a gel box, and fluorescent proteins. The rest is the same. And the fluorescent dyes include, so these fluorescent dyes, cyanins, they are cyanin 2, CY3, CY5. Immersion system. So immersion system is a system which use these dyes, and they're actually patent. Okay, so CY2, CY3, and CY5, they are protein stains of the same size. So if we use CY2 with one protein sample and CY3 with other protein sample, so this dye will be of the same size, so they will not hinder the protein migration actually, because the size of these dyes is same. And these have similar sizes and charges, which means that individual proteins move to the same place on 2D gels, no matter what dye they are labeled with. So the size of the, most important is the size of these dyes will be same. And detection between, they are sensitive enough, and they can detect up to 125 picogram of a single protein. So is it clear up till now?
Yes, sir.
So who will explain? Fatma Mohsin. फातिमा, आवाज आ रही है आपको? Umema is there. Umema, can you hear me? Zumar, आपको आवाज आ रही है? जुमर, आवाज आ रही है आपको? Gee, you raised your hand. Okay, you have problem with the mic. Okay, and Suhaib, is here. Suhaib, can you hear me? So, and Simal, Sibga, Safgar, you there? Hamza Nadeem. Hamza? Hamza?
जी
जी
सर, अस्सलाम वालेकुम।
वालेकुम अस्सलाम जी। ये स्लाइड पे क्या पढ़ा है वो बताएं।
तो सर बेसिकली इसमें भी जो अभी हमने पहले डिस्कस किया था SDS पेज का प्रिंसिपल,
हाँ
वही जो है ये डिफरेंशियल जेल इलेक्ट्रोफोरेसिस में भी यूज होता है। और सर ये भी 1 और 2 डायमेंशंस के अंदर जो है यूटिलाइज की जा सकती है। फिर सर बेसिकली इसके अंदर भी जो है फ्लोरेसेंट डाइज जो है वो यूज़ की जाती हैं। उसके आपने नेम जो है यहाँ पे लिखे हैं CY2, CY3, CY5। और सर इसके अंदर मेन चीज़ जो थी वो यही थी कि जो साइज और चार्जेस होते हैं इंडिविजुअल प्रोटीन्स के वो सिमिलर ही होते हैं, और ये बेसिकली जो मूव करती हैं वो 2D जेल के अंदर ही मूव करती हैं, जो मर्जी आप डाई यूज़ कर लें। तो ये जो चीज़ थी इसके अंदर और सर एक और चीज़ थी कि जो सिंगल प्रोटीन है उसकी जो डिटेक्शन होती है सर वो 125 पिकोग्राम तक हम कर सकते हैं।
जी एग्जैक्टली। अच्छा, वन थिंग वी नीड टू नो कि these stains are fluorescent stains, fluorescent protein stains। इससे पहले जो stains हैं वो फ्लोरेसेंट तो नहीं थी ना, दे आर फ्लोरेसेंट। वो जो कमर्शियल ब्रिलियंट ब्लू और वो है वो those are not fluorescent, but they are fluorescent। ठीक है, so they are and silver stain, so these are actually fluorescent stains।
सर
जी जी।
सर ये जो फ्लोरेसेंट डाइज होंगे ये जो प्रोटीन के स्ट्रक्चर में किसी स्पेसिफिक अमीनो एसिड्स पे जाकर बाइंड करेंगे या
Yes,
बॉन्ड
they will bind to specific amino acids. We will be talking about in the coming slide।
ओके सर।
ठीक है। So so these differential gel electrophoresis, okay, so let me check the chat। Okay, so these differential stains, they contain different stains, and these proteins, the protein samples are produced by homogenizing cells. But the cells are homogenized, means they are lysed and they are homogenized. Standard chemistry links dyes to proteins via lysine side chains. So lysine side chains will be used, their amide bonds will be used to link the dyes, actually the fluorescent dyes. So lysine side residues, their amide bonds will be used to link the dyes. And linear response to protein concentration over of 10 raised to power 5 concentration range. So it has a linear response, means more the dye, more the concentration, more will be the dye that will be attaching to these proteins. So if you can see here, this NHS is NHS is N-hydroxysuccinamide, and this is the structure, and this is the functional group, the NHS ester, and this is the reactive group shown in green color, and this is linked with this dye. So this could be sign in 2, 3, or 5. So it is how it binds with the protein, the amide group of the protein. That will replace this NHS ester, and that will bind it with the dye. You can see here, and here in this diagram, the R is the labeling reagent. R is the labeling reagent, or one end of a cross linker between the NHS, have the cross linker between having the NHS reactive group. So this R could be, like we already talked about, R could be CY2, CY3, or CY5. And these are actually the fluorescent dyes. And here the protein, the primary amine on protein. So the primary amine on the protein here, the primary amine on this protein. So now this they will react, and this is a stable conjugation. Amide bond is formed between this reactive group and this protein. So this amide bond is being produced and the NHS is being released. Am I clear?
Yes, sir.
Okay, Kalimullah.
यस सर
जी आप इसको बताएं को।
ये बताया गया है कि ना जो DIG है जो जेल इलेक्ट्रोफोरेसिस है ना इसमें पहले हम सैंपल जो होता है प्रोटीन सैंपल जो हम जो वो कैसे लेते हैं वो हम होमोजेनाइजिंग करते हैं सेल्स की तब हम प्रोटीन सैंपल लेते हैं फिर हम डाइज यूज करते हैं वो डाइज जो है वो अमाइड बॉन्ड के जरिए अटैच हो जाती हैं फिर जो है कोई क्या हम देखते हैं कि जो रिस्पांस है जो लीनियर रिस्पांस होता है जो प्रोटीन कंसंट्रेशन है उसी के हिसाब से आते हैं डाई उसके साथ अटैच जितने कंसंट्रेशन ऐसे डाई अटैच हो जाती हैं उसके साथ
गुड ग्रेट ग्रेट और कौन इसको फर्दर एक्सप्लेन करेगा Fatma Ramzan. Fatma Ramzan is there?
Yes, sir.
जी बाकी जो बात कोई बात रह गई हो इस स्लाइड पे कैन यू एक्सप्लेन दैट और थोड़ी सी एक्सप्लेनेशन
सर इसमें यही है कि जो डाइज हैं वो एस्टर के रिएक्टिव ग्रुप के साथ जाके बाइंड करते हैं और फिर जब प्रोटीन प्रोसेस होती है तो डाई जो है वो अटैच हो जाती है और फिर डाई से ही हम उसकी लीनियर रिस्पांस चेक करते हैं कंसंट्रेशन उसकी कि जितनी प्रोटीन की कंसंट्रेशन होगी उतना ही डाई फ्लोरेसेंस करेगा
ठीक हो गया और ये अटैच कहाँ किस तरह होगा फ्लोरेसेंट डाई like how the reaction will occur with the NHS ester जी
सर कौन एमाइड बॉन्ड बनाएगा
जी बिल्कुल बनाएगा किस तरह बनाएगा ये डायग्राम में शो हुआ है ना दो डिफरेंट पर्सपेक्टिव में शो हुआ है डिफरेंट एंगल से अच्छा चलें जी जी किन्जा हाँ किसी और से पूछते हैं तो अगर वहां भी क्लियर ना हुआ तो फिर करेंगे दोबारा एक्सप्लेन इसको किन्जा इज देयर किन्जा
सर सर ये डाई है ये अटैच होगा क्या नाम है NHS एस्टर रिएक्टिव ग्रुप पे अटैच होगा
ठीक हो गया ये ठीक है
सर इसमें ये माइक्रो इसको रिप्लेस कर देगा उसके बाद हमारे पास डाई आ जाएगा फिर वो फ्लोरेसेंस देगा
ये इसके साथ अटैच हो जाएगा विद एमाइड ग्रुप और यहाँ पे ये जी जी एग्जैक्टली और यहाँ पे इस डायग्राम में क्या है
सर इसमें ना हमारे पास NH एस्टर अटैच होगी प्राइमरी अमाइन प्रोटीन साइड ठीक है और उसके बाद pH हमारे पास 7 to 9 होगी फिर हमारे पास ऐसे रिजल्ट स्टेबल कॉन्जुगेट अमाइन बॉन्ड बन जाएगा और हमारे पास NH हाइड्रोक्सी अमाइड आ जाएगा
ये रिलीज हो जाएगा and hydroxysuccinamide जो है that will release and we will get the fluorescent dye attached with the protein and कहाँ पे ये attach होगा lysine side residue के साथ with and that the amide group of that so lysine residue इस protein में जहाँ भी होगा तो lysine उसका side residue amide group के साथ ये reactive group attach हो जाएगा is it clear
yes sir
so the pH range may vary from 7 to 9 here is the pH 8.5 and so that can vary Fareen is there Fareen
yes sir
जी आप एक्सप्लेन कर दें इसको थोड़ा सा
सर डायग्राम को एक्सप्लेन करते हैं सारा
जी ये सारा ही कर दें
सर इसमें सर ये बताया कि प्रोटीन सैंपल्स है ना सर ये होमोजेनाइजिंग सेल्स में प्रोड्यूस होंगे और सर इसमें डाइज का बताया कि डाइज हम कैसे यूज करते हैं तो सर फ्लोरेसेंस प्रोड्यूस करने के लिए सर अब इसमें बताया कि जो डाई है उसके साथ NH एस्टर जो होता है वो अटैच होता है और फिर जब सर हम उसमें उसको जब प्रोटीन से इंट्रोड्यूस करवाते हैं तो जो प्रोटीन होती है वो डाई के साथ इंटरलेट मतलब हो जाती है। तो सर नेक्स्ट डायग्राम में बताया कि कैसे होती है। जो एस्टर रिएजेंट होता है सर वो प्राइमरी अमाइन के साथ जो है वो रिएक्ट कर जाता है और NH जो है सर वो रिमूव हो जाता है और हमारे पास कॉन्जुगेट स्टेबल मॉलिक्यूल आ जाता है अमाइड में।
Great, good हो गया, good हो गया. So, class, any question? I want to stop it here because we have a few more slides, but I want to focus those slides in the coming class.